Hello, welcome. Good morning, actually. I'm broadcasting live here from Park City, Utah, which, as many of you know, I've been spending a lot of time out of New York City, enjoying glorious Park City. Thank you for joining us today. And John, are you in Princeton? I know historically you were in Princeton, but in New York City?
I'm still very close to Princeton. I'm not in my New York office, in my home office today. And that's in a little town called Cranberry, New Jersey.
Yeah,
which is very near Princeton.
Yeah, I used to play golf at Yasna Polana over there in Princeton.
Oh, right? Yeah.
Yeah, the Johnson & Johnson folks. So welcome, everybody. Thanks for sharing your day with us. We're going to be chatting for just about an hour. Very, very interesting topic, and I'm grateful to have Dr. John Hall with us today. As you well know, we can't hear or see you. We know you're there. And if somebody wouldn't mind just saying hello in the chat box so we know we're getting out to you, that would be helpful. Even though we can't see or hear you in the context of this Zoom, we encourage your questions. Please post them as they come to mind. We have quite a few folks coming on today, so we'll get to as many as possible. We're going to be recording this, so during the presentation today, if you think of anybody that might benefit from knowing, they can either listen to the recording that we'll post on the Family Office Insights YouTube channel, and then we're also going to send it off to John as well, and he can post it where he likes. And further to that, we'll can make sure that everybody gets in direct touch with John if it's of interest to you. And of course, the purpose of today's call is to see if anyone in the community is interested as an investor in this cancer drug technology and patent, specifically the patent you see on the screen there. So again, super grateful for Family Office Insights members' participation as usual. You guys are just awesome, and I appreciate how much you participated in our webinars. And for the record, we're going to be doing some in-person gigs in New York City, gearing that back up and see how that goes. So welcome. And here we go. John, take it away.
Hello, everyone. First, welcome and thank you for attending. My name is Dr. John Hall, as you already know, and I am the Vice President for Research and Development at a small biotech firm located in New York City. That would be Molecular International Research. On the screen, you can see that I'm going to tell you about an investment opportunity in new drug development based on our recently allowed patent, which is titled Selective Anti-Cancer Agent Effective for Prevention and Treatment. Now, we all know, I think, that cancer has a profound impact on the lives of those who succumb to the disease. But it also has an equally powerful impact on the families of the patients. So I'm thinking I may be in the right place presenting this appeal before Family Office Insights. And my goal is to generate interest, as Arthur just said, and find among you those who would consider further conversations with the possibility of making an investment. So let me start out by just summarizing what I'm going to do here. So I'm going to tell you about this new drug candidate. I keep referring it to a drug, and you'll see me keep doing that, but it's still not approved by the FDA as a drug that can be sold and used in humans for their cancer. But it has been researched. It is patented, it is non-toxic. And I think I'm going to come back to that, of course, because this is a major advance in biomedicine and in cancer therapy. We found that it's effective against most types of cancers we've tested, including those that are difficult to treat. The notorious pancreatic, lung is in that category, and we're considering some others. Everything we have, all of our data, predicts that the drug will be safe in the human body. I also will describe the anti-inflammatory function of this drug. It's a second application, and it opens another market, which is significant. So I think I can say, taken together, the advances of this drug make it an outstanding investment opportunity. What am I asking for? $3.2 million to fund the transition to human clinical trials. So the goal specifically is submission of an investigational new drug application called an IND to the FDA. That's the Food and Drug Administration, which controls and regulates the development and approval of drugs. The IND is what enables the human clinical trials essential for ultimate FDA drug approval. Completion of the IND research will strengthen opportunities for licensing or acquisition by a partner in the pharmaceutical industry. So the IND research, which is really what it is focused on toxicology studies, which is the main requirement for the IND application, showing that our agent, our drug candidate, is safe enough for use in human clinical trials. There are also some other research components of the IND application, which I'll talk about a little bit later. So I want to give you an overview of the process of drug development, so you can see where we are in it, what generally is done. This pathway is pretty well established and pretty well trodden. It starts with research usually, and if that research in cell, molecular, animal studies, if it's successful, there can be an invention, a new observation, a new drug target, a new mechanism of action. So in our case, we have a novel anti-cancer and anti-inflammatory drug candidate. We have been successful in patenting this. The patent includes details of the invention and claims covering compositions and methods. The next step, which I've thrown in because it's so important, is valuation of the patent. If it's going to be marketed, if this technology is going to be successful, we have to have a benchmark. What is it considered worth? And we've done that with a reputable firm, IP Metrics, and that initiates the marketing phase, which is really part of what I'm doing right here. I'm appealing for funds to do the IND, and that will, as you'll see, the valuation of the patent is important in this marketing effort, which also includes outreach to pharma, which we've done a couple of conversations we've been able to have, which help us understand what we need to do at the IND step in order to enrich this opportunity in the bigger pharma market. So the center of the slide here is IND application to the FDA. That's the Investigational New Drug Application I just spoke of, and that's where we are. There is no way forward. Down at the bottom, you can see FDA approval of new drug. That's not going to happen without the IND, a successful IND application. And then critically, the human clinical trials, which are meant to establish safety for the drug in humans and efficacy, which means for anti-cancer drugs that it actually reduces, say, the tumor. And the goal of the FDA, their main obsession is trying to have a safety value that's significant, but they also need to see that the thing works. Okay.
Hey, John.
So, yes.
Can I just jump in for 1 second?
Sure.
Back to that other slide. For those of us that's not, then this is super helpful, by the way. If for whatever reason their INDA application isn't procured, and I don't mean to put negative things out there, but would you pivot and go to other countries, or is there value in the patent independent of its FDA approval?
I think if we want to make a drug, and we do, FDA approval is essential.
Got it.
I think what you've just raised is worthy of consideration. And I don't think we've done much of that because we're so focused on this IND and ultimate clinical trials, FDA drug approval. But I want to say that we have robust confidence because our own safety profile for this agent is it's non-toxic. So if the FDA stands by its claim that it wants to show safety and it wants to approve, even for an IND, a drug that's not toxic, we can meet that requirement. We have confidence there. So we think this system is going to work in our favor because requirements for IND we can meet, and we can expect firmly to pass the test in the IND, to pass those formal toxicology studies.
Yep, totally makes sense. Thank you.
Okay, sure. So let me back up a little bit and just talk about what problem we were trying to solve when we started this. The problem is that chemotherapy drugs are not selective for cancer. To be brief, cancer cells grow fast and chemotherapy kills fast-growing cells. But chemotherapy also kills normal cells that are fast-growing. The death of these normal cells causes severe side effects, and patients suffer, and treatment must be stopped before therapy is complete. And I think Arthur and I were talking just before we came on. I can say this is common knowledge. People know people, if they haven't themselves been in some chemotherapy regimen, how devastating the treatment itself can be. And people are sometimes wondering, and out loud, that the treatment is in some cases just as bad or worse than the disease itself. So this is a formidable problem that we address here. And let me tell you what we came up with, what the answer is. The answer is a selective anti-cancer treatment. And selectivity, I'm going to use that word a few more times because it's so important to the identity of our drug. And as this text says, this patented drug, it's called diethylflavopiravine, selectively destroys cancer cells in a solid tumor or at a site of metastasis without damaging healthy tissue. So it also can be used for cancer therapy for many different cancers. It's a broad spectrum agent, which is of course advantageous. It can be administered orally, which is a great route of administrations. It avoids the complications of IV, intravenous administration, as well as the cost, and makes it more amenable to human use. You can pop a few of these pills as prescribed and scheduled by your oncologist and not have to go into the clinic specifically for administration of the drug into your veins. It is well tolerated at large systemic doses. That's the message about lack of toxicity. But it has a second application, as I already said, as an anti-inflammatory. And this is it's good to have more than one. Cancer is what we focused on, but we've learned in the course of doing this that the molecule has very significant anti-inflammatory effects, and I'll come back to that in a bit. Well, so why is it so special? As I've said, it's non-toxic. And I refer to that as the gift that keeps on giving. And that means we redefine the anti-cancer treatment cycles, which I just described a moment ago. Normally, these drugs are given for a while. They're prescribed. There's a schedule. It's perhaps a period of a few weeks. The drug is administered. And then there is a respite, a withdrawal of treatment so that the patient can recover from any side effects, literally from the toxicity of the treatment. And then after a certain point, the patients say their immune system cells, their white blood cells have come back, then another round of treatment begins, and that has a duration, but that's also limited because it's not possible to give these drugs continuously. All right. I mentioned that it's broad spectrum. It kills cancer cells of all types tested to date, including hard to treat. Well, I want to finish one point about the lack of toxicity, which I was just saying. With our agent, we predict that this drug can be given by the oncologist according to optimal schedule for the optimal amount of time. And a key point here, really the key thing here is that the patient gets a respite when the treatment is suspended for a while, but so does the tumor. That tumor can also recover and start regrowth. And in many cases, that's what happens. The tumor takes advantage, the cancer takes advantage of this cycling of treatment to regrow. And what happens is the regrowth can sometimes turn more aggressive. So there's an advantage to starting and maintaining the anti-cancer treatment until the desired goal is reached, or until you determine that you have to bring in another drug, for example. So it's broad spectrum. I've covered that. Another thing very important to us from the standpoint of the use of this in the clinic, but also from the strategy of getting interest, is working together with the drugs that are out there. And we've tested our drug with several of the common chemo drugs. And these combinations are very, very effective. So for example, gemcitabine, which is commonly used for pancreatic and lung cancer. We combine with that drug, and you put those two together, you can kill 100% of the cells. It's also an anti-inflammatory. This is indeed a different application. And in the valuation we had done, it was treated as an independent application. And it has its own value here because we see potential to treat the now well-known cytokine storm, which is a result of persistent COVID-19 infections. And in fact, as you may know, most of the patients who die of COVID infections do so because their own immune system is so overstimulated that this immune cells that are supposed to attack the infected cells attack neighboring healthy tissues. And that's the main issue there. So attenuating these immune signals that's bringing about this storm is a very important function or application for our agent. And it's also, as you'll see, I have some data towards the end if we have time, but this anti-inflammatory effect is a source of positive side effects instead of negative side effects. So for example, if you're protecting tissues from damage from an inflammatory condition, then when you take this drug, the patient takes this drug for a cancer, they're also getting tissue protection from damage by their own immune system. So the idea of positive side effects is worth mentioning. It also targets cancer stem cells. I'm not going to talk much about that, but cancer stem cells are a form of stem cells, not the healthy ones used in therapies and renewal of tissues, regrowth of cells in the body. But these cancer stem cells are literally cancer cells that have hijacked the mechanisms of regeneration and persistence that normal cancer cells have. And they're responsible for cancer recurrence and metastasis, cancer spread. So there are other advantages. No time for that now, including oral administration. It's well tolerated, and so on. Well, I want to be upfront here about the risks of investment, because we have a very high quality drug candidate with the incredibly desirable aspect of being non-toxic, of also being selected. Those things really mean the same. Our drug goes after cancer cells, not healthy cells. But whatever, 10% of drugs that are tried and undergo years of development, only 10% of the drugs that undergo this process, which I was just describing in a previous slide, the process of drug development, only 10% actually reach the market, which is certainly not a high success rate. And unfortunately, these failures often show up in human clinical trials, sometimes in late stages where there's enough people in the trial so that the side effects that do occur can be seen at their real frequency, and issues about efficacy can be assessed. So overall, up to 50% of these drug candidates lack the efficacy that was expected or predicted. A third of them are roughly because of unmanageable toxicity. And then there's poor pharmacokinetics, which just means that the drug doesn't end up where it needs to go to help kill the tumor, to shrink the tumor. The body may recognize it, and it may, of its own biochemical interactions in the body, end up in tissues that are not relevant to cancer treatment. And then, of course, there's a problem of commercial interest. Somebody who's got a good product has to be able to sell it to the industry and make it into a drug with the support and the funds of the pharmaceutical industry, and that's not trivial. Okay, let me just show you the molecules. Diethylflavopiririne is a derivative of a naturally occurring molecule. And I could have titled this slide, Improving on a Natural Molecule. So flavopiririne is on the left. It is a carbon-based, a ring-based structure. You can see at the apex, all those apexes, there's a carbon atom, there are hydrogen atoms also. And then here at the apex is a nitrogen, and here's another nitrogen. This ethyl group hangs off the side of the molecule here, and that apex means there's a carbon there and there's a carbon at the end. What we did is remove that group, and we end up with diethylflavopiririne. It's really the same structure, except that side branch has been removed. Now, the point is that the naturally occurring flavopiririne is not patentable. The newly synthesized molecule we've made, diethylflavopiririne, is patented. It has been patented, and that's vital. That's necessary for development. But we have the advantage of flavopiririne, the ancestor, the natural molecule, having been researched to a considerable degree for its anti-cancer activity. And lo and behold, our molecule maintains that activity. So it's a different structure, only slightly different, but that's an advantage. But it maintains anti-cancer effect. And this is critically important. That's not surprising from a chemical standpoint. The molecules are so similar that the anti-cancer effect isn't conferred by this ethyl group that we've taken off. You can remove that and still keep anti-cancer effect. So diethylflavopiririne is known as NT1391. It's just the name. NT refers to the non-toxic. It's important enough, and in our safety profile, it's reproducible. So it's called non-toxic 1391. And that 1391 refers to the fact that it's the sulfate salt of the compound. It's just a variant which is associated with the sulfate group, which makes it stable for use and administration.
John?
Yes.
So is this natural occurring compound? Is that the right way to, is it a compound?
Yes, it is. Sure.
Is it successfully used in the treatment presently of cancer?
Yes, it is, in the sense that it is the primary anti-cancer compound in an extract prepared from the bark of a tree that grows in the Amazon rainforest. So the natural history of this, it's one of those naturally occurring products that has been studied for its anti-cancer effects. And it's also very low in toxicity. I didn't mention that. It's not like by taking the ethyl group off, we got, whoa, now it's not toxic. The starting material, flavopiririne, is low toxic to begin with. All we did was move further, lessen that toxicity even more. So yes, indeed, flavopiririne is used against cancer. And there have been, recently, there's been a flurry of activity with this molecule, with the purified compound flavopiririne in a number of studies for breast and I think liver. And there's another cancer, which shows that it's effective.
And being that it's not patentable,
Mm-hmm.
I understand the commercial implications of taking that red piece off and now it's patentable.
Mm-hmm.
Has it been commercialized such that it's widely used?
No, it's not.
Yeah. And
that's one reason why our patent has value, because we're the ones who've highlighted both the consistency of its anti-cancer effect expected in the structure, based on the chemical structure, but also we showed that compared to flavopiririne, it's less toxic, which can only be a good thing if you're going to give it to humans. But that has not been done. So I think it's our research and our patent that is the first to make the case that this is a synthetic molecule, and has been patented, but that it has anti-cancer activity and can be treated as a drug once it's allowed. It can be commercialized because of the status, but it's also even less toxic than the naturally occurring compound. That's why I said this is like improving on a natural molecule. And people, there's a tendency to think if it's a natural molecule, it's good. It's not too toxic. But here, the synthetic one we've made is an improvement.
Is it an improvement by 10% or 20% without putting you into the corner? I know you're a
I'd have to go
scientist.
back and look at the data. We saw a significant increase. So one of the things we did was use an animal model, which is essential for all of this research, and give the mice a purified flavopiridine and another mouse, the sibling, a purified diethylflavopyridine, and see the effects. And the answer is you could give significantly more by diethylflavopyridine. Yeah, it's very interesting because, in fact, flavo, as I said, is not toxic to begin with. So we're way out on that vanishing toxicity from our experiment.
Thank you for
that. Yes,
that.
and that's really what this whole presentation is about. What we need to do is go confirm that in the formal toxicology studies, right? That's really the challenge of the IND is, okay, you have this data, looks non-toxic. Let's make sure you're right by doing study not just in mice, but in larger mammals like rats and dogs. And let's find out what the real toxicity is in an independent lab that is a reputable lab that knows how to do these experiments.
So I don't want to jump ahead, but the pedestrian thinking that I have, being not in this arcane subject, is then, okay, so that's cool. Let's accept for that it's super non-toxic.
Okay.
How effective is it?
Well, our data show it's very, and if we get to the data, you'll see an example or two.
Yep, I don't want to slow you down.
Okay, no, I'm glad for the questions. Very helpful. So well, let's move on to what exactly effect does this have in a cancer cell? When NT1391 enters a cancer cell, what is the result? What's the end result? And the answer is selective induction of apoptosis in the tumor cell. And apoptosis is a pathway that's built in. It's called a program, but it's built into our cells. And it is a pathway that leads to self-destruction or cancer cell death by suicide. This is a remarkable thing in biology, and it occurs during development when certain cells are no longer needed in the maturation of the body plan. They go ahead and commit suicide. So they're removed from the body. And that's a program, it's a great program to manipulate if you're going after a cancer cell. If you can selectively induce that result, you're killing a cancer cell. And if it's indeed selective, you're not going to do that to a healthy cell. And we have our data in the patent shows apoptosis in pancreatic cancers and in colon cancers. Those were the two models we used. And it turns out NT1391 can induce apoptosis using a number or multiple signaling pathways. And I like to think what the message of this is, once this molecule's in a cancer cell, that cancer cell's going to die by one of these pathways, one way or another, one pathway or another. The end is there for the cancer cell because it seems to be so flexible. It'll use one pathway, and if that isn't either doesn't work or it doesn't stimulate sufficiently, it'll find another pathway. So it's impressive biochemical activity in the body with all anti-cancer effect. I'm just showing you a front page of the patent so you can see it. There's really not much for me to say about this. For some reason, I wanted to keep that in there. And let's go on. Yeah, the patent, at least at this point, just to give you an idea of what's claimed in that patent. And first is a therapeutic method, administering the 1391 compound in one or more doses to a subject at risk for cancer or afflicted with cancer. So it's a prevention and treatment claim. I also mentioned combinations with other drugs, and that's claimed in the patent. It's a method comprising co-administering our drug sequentially or not with a chemotherapeutic agent other than this our compound, wherein the chemotherapeutic agent is also active in the treatment of cancer. So we're covering this combination potential. And then finally, a method for treating inflammation and preventing resultant tissue damage, comprising administering one or more doses to the subject in need thereof of the NT1391 compound. It's in an amount effective to reduce inflammation and thereby prevent tissue damage by infiltrating leukocytes. So there are three key experiments that back up those claims. One is the anti-cancer. So the drug candidate shrinks pancreatic and colon cancer tumors in animals by oral administration with no detected toxicity. It's effective in drug combinations, and I mentioned the combination with gemcitabine, which is extraordinary, but it has the ability to synergize with other anti-cancer drugs, which, as I've said, has real value for both cancer treatments and for the marketability of our product. It's an anti-inflammatory, which prevents tissue damage caused by persistent immune response, and we have data showing how that works. Well, let's just go back and make a few points about the IND, give a few details about this process. As I've already said, I think the total cost is going to be 3.2 million. And this is what allows human clinical trials, which are essential to get drug approval. And toxicology is the centerpiece. I mentioned rats and dogs. It includes tests for impact on cardiopulmonary function. And we have designed metabolic and toxicology studies with Charles River Labs. And in the conversation there, the chief scientist said, if you're seeing efficacy for pancreatic cancer by oral administration, you're really onto something. I thought I'd quote him here for that. So the IND application is a process we've also been through before with our RNA patent. The one that, this was an earlier patent, which is an immune support RNA fragment, which we completed the IND application process with the expert assistance of Dr. F.A. Hoffman, who, and this is great profile for her, she used to work at the FDA and has extensive experience with successful IND applications. And we, as I was just saying, previously compiled a complete IND application for RNA-based drug candidate, working closely with Dr. Hoffman. So it's not our first time trying to do this process. The collaborators, just to show you, patent valuation is IP metrics. Worked very closely with Fernando Torres, who was extremely helpful and really took the time to become familiar with this technology and got insight in, and was able to see how stunning it really is. The compound synthesis, this is a synthetic compound. One of the things that we have to do to move forward is make a lot of it so we can do the toxicology studies and ultimately get ready for clinical trials. That's APAC Pharmatech. We've worked successfully with them for some time. They're reliable, they make the product at very high purity, and that's who we've chosen to make the compound for the IND.
Is there a supply issue, being that it's a bark from a tree in the Amazon?
Okay, that's the naturally occurring, right?
Oh, I see.
So
okay.
That's, yeah, no, you've hit on an important issue. That's a sustainability. That's a fabulous tree. It's a Pau Pereira tree, and the Pau Pereira plant extract is an effective anti-cancer agent. And indeed, our CEO of Molecular has a separate company that prepares that extract. And I have helped, as a director of the foundation there, show many experiments showing that that does work as an anti-cancer. So it's proven. But there is a sustainability. You can only get so much of this bark. The bark does regrow. So when it's harvested, the tree can survive and grow the bark back. But the whole advantage of a synthetic compound is that you can make as much as you need.
Yeah, just create it. Yeah.
Yeah.
No.
So for toxicology, it's Charles River Labs, and I was just talking about Sam Chang and Kyle Van Ripper. And Freddie Ann Hoffman at Heterogeneity is the IND application. And she also does the filing. So we can get the thing done by an expert. So how does the cost break down? I said 3.2 million. The compound synthesis is 865,000. I have insisted that we make enough of that, not just to complete the toxicology studies, but to have some left over in case it's needed, but also to make it in a form that is acceptable for use in human clinical trials. So if there is material left over, we won't throw it out. We can take advantage of using it in the next step, which is in humans. The toxicology studies, that's rather elaborate. I'd love for you to look over the research plans. There's a lot in there, and it's not just giving the material to animals. It's for example, giving it to an animal not to look for anti-cancer, but to see how the body metabolizes it. What happens? Where does it go? And there are two questions there. Does the body break it down? Is it changed by digestion, for example, or other enzymatic activity? Or is it intact? Our evidence says it stays intact, which is a benefit. But pharmacokinetics is the process of determining where in the body the compound localizes. And that's important because in those tissues where it localizes significantly, you have a greater concentration to deal with a cancer in that tissue. And so, for example, lung is at the top, but colon, kidney, spleen, all the way down to pancreas, there's considerable localization. So those are cancers that we feel confident being able to treat. I also want to add a budget for molecular mechanism of action. And that's a detail I'm not going to get into today, but just to say, we know what we're doing as an end result is inducing apoptosis. You recall this suicide program we can induce in a cancer cell. But we need to know a bit more about what the molecule is doing in the cell to switch on those apoptotic pathways. What is the binding target, for example? IND application and FDA filing, that's Freddie and Hoffman's work, and there's some additional funds in the budget. So that covers what we're going to do with the money when we get it. And drug development, the valuation is very valuable, and those of you interested will have access to it. It specifies the timetable and the costs, overall costs of drug development. The IND phase, which we're so focused on now, is vindication of our safety profile, metabolics, I mentioned, pharmacokinetics, analysis of drug target interactions, and optimization of drug delivery. We can get that done. We folded that in to the IND research. Then there's FDA approval for the investigational drug application. And then finally, clinical trials, which is what we need to get to. We foresee 3 tracks: pancreatic cancer, by fast track, that's another advantage. There's so few agents of any kind that actually are effective against pancreatic cancer that the FDA will allow a fast track development, which means you can go through the process of the clinical trials and approval more rapidly than if you just have yet another candidate for another anti-cancer, for another cancer. Lung cancer is another application. Fast track may also be possible here. We're not 100% sure, but not much works for lung cancer. It's still very common, and surgery is really tough there because it's such a delicate tissue. And the drugs haven't been working very well, the other drugs. So I think we have a real in there. And then third, inflammation application. That's cytokine storm, but there are other inflammations that can be treated. So then you get FDA approval of a new drug. And that's what we're seeking. And then finally, let me go down again, sorry. Yeah, if we get to the end of this, FDA approval will yield an anti-cancer drug with a new level of safety, and drug action won't depend on the mechanisms that have been tried and have failed for so long. So the fact, remember we started out talking about rapid cell division. That's not a selective target. That's not the way to go. Mutations in patients' cancer cells, that's not our target either. We're not dependent or strictly dependent on the genetic profile of changes in a cancer cell. There are many of them, and many of those genetic mutations are important for cancer progression. But we're going after, I would argue, a more fundamental target. The drug will generate a new platform for drug combinations. I've mentioned that, and will improve many forms of treatment. And just by way of an example, I was speaking a few years ago to an immunotherapy, a new guy who's working with immunotherapy, which is a method for turning our own immune system and focusing it on the cancer cells. So we use our immunity to take out cancer cells. And he told me how badly he needs a drug that could shrink his patient's tumors by apoptosis so that immunotherapy could become a more effective treatment. He might as well have been describing our drug, and many oncologists could say the same for their goals with their patients. So our value is that our product is, I believe, what the cancer treatment industry needs. And of course, we feel an urgency to move forward. Here is one example of the pancreatic, this is pancreatic cancer application, which says the patent is valued at $278 million for that application alone. And this is with a conservative assumption of a 15% market share. So we're not going in there, and this valuation doesn't say we're taking over the world and going to be the drug that everybody uses for pancreatic cancer or any other cancer, but we're going to have a significant market share, and there is quite obviously significant money to be made. Lung cancer, for which a novel therapy is also urgently needed, is the second application with an even higher fair market value, reflecting, in fact, greater incidence than pancreatic. It's broad spectrum and selective anti-cancer action of NT-1391 supports extensive applications in oncology. It's be great for colon cancer, for example, as well as important contributions to treatment of inflammation, and the details are in the summary and full report of evaluation from IP Metrics. The lesson learned is that NT-1391 will be an extremely profitable drug. And then if you look at the big picture for all three, and this is not the full extent of the applicability in cancer, but if you add that up, the lung, pancreatic, and inflammation and their contributions, the total is 799 million, rounding up a little bit.
What's the time frame for the first drug?
To make this?
No, I'm sorry, I'm not clear. You raised the 3.2 million.
Yes.
You flip the switch, you start all that process happening. How long does that process take, the IND?
We can do that in a year.
And then what?
And then we get the IND approved, and then we set up clinical trials.
And how long would that take?
Those take a few years.
Yeah.
And I think it's a time-consuming process.
Yeah.
And that's part of what we live with.
Yeah, no, I've heard that it's just is what it is, right? So
that's right. That's
yeah.
right. I think, as I've said all along, we have some advantages. If we get that low toxicity confirmed, that's a leg up in moving forward.
Yeah.
And so we have to see.
Does the fast tracking apply to the human trials as well?
Yes, that's exactly what it applies to.
Yeah.
That's right.
And so just without having to be precise, do you have to raise another $30 million to do the clinical trials?
What I anticipate is the license or acquisition by a pharmaceutical company.
Yeah, so I thought
that's the exit. That's where this
yeah.
belongs.
Yeah.
It's going to be a drug. Let the experts take it over.
Yeah.
And
let you guys take the risk, you prove it out, and then they'll write the check, right?
it does come down to that in a fundamental sense.
Tend to oversimplify things, John, as you
no, but that's right.
Yeah.
Yep. Okay.
Yeah. Continue.
I think, I want to tell you that our team, that's Sylvia Beljanski, who I spoke of a little while ago, President and CEO. There's me, VP, Research and Development. Sylvia has a very interesting background. She's born in New York, but actually grew up in Paris, where she was educated, and became an attorney, admitted to the bar there, and then decided to focus her efforts completely on developing the plant extracts that her father had initially done as a scientist at the Pasteur Institute in Paris, France. So the Pau Pereira extract I spoke of was discovered by Mirko Beljanski, her father, and she's taken on the job of producing it and selling it. I joined in 2004. We have new intellectual property as a goal. Together, we have 3 patents: prevention and treatment of prostate cancer. A second is the RNA therapeutic treatments based on administration of small RNA fragments. And this new patent is the subject of today's presentation. The company is privately held, and there have been no outside investors to date. That's what we're searching for now. The IND phase drug development is the immediate need, and completion of drug development will be funded through licensing or acquisition by a pharmaceutical. So that's Arthur's question. That's our plan. This goes over what I was just doing. You can see my background. Princeton University, New York University. I was on the faculty at Rockefeller University in New York. And I did start my own biotech company some time back before joining Molecular International. We also have some really great attorneys who are part of the team. Gary Tanagawa has a PhD with really impressive work at MIT and Harvard Medical School. He did a postdoc at Memorial Sloan Kettering Cancer and then transitioned to the law at Georgetown. And he's been a great help in putting this patent together. It's really a wonderful contributor. Josh Goldberg is newly with us. He has done a lot of deals. He's a deal maker, and he can broker the kinds of deals we need. Focused on both acquisition of worldwide patent portfolios, particularly in the pharmaceutical and biotechnology industries, as well as assisting his clients with licensing and commercialization of such products. So that's the presentation. I'm happy to go on and show a little data if we have time. I'm not sure we do, so please let me know.
Yeah, we want to be polite to the group, but
Yes.
a couple questions came in on the side door here. Those 2 groups are the 2 attorneys that are doing the patents for you?
That's right. Gary Tanagawa did this new patent. He and I worked very closely on it for some time, and he brought a lot of his expertise. And Josh is in a position to help us with the negotiations for, say, forming getting an investment and nailing that down, papering it, so to speak. He has experience doing that.
And also in the context of the acquisition by the pharmaceutical company, his role there?
Yes, he's in a position to help us get the terms and negotiate and establish, protect our interests. And still, as you know, it's a give and take. We've got to try to provide what we can, but also get what we deserve.
Yep, of course. I forgot to put my video on. What else can go wrong here, John?
I've been thinking about that. I think about it because you have to, but I think it has to be something that's completely out of the blue, something completely unexpected that we can't anticipate. And that's where we are. And I want to say, for those who are interested, I would like to have a further conversation to that point, because there's reasons for our confidence, which are in some of the data I don't have time to present today. But there's no reason we have now to worry about why it won't work. But as you know, it's a human endeavor, and however the excellence of the product, something can go wrong that we don't even imagine. That's the answer.
Is it fair to ask to compare the commercial success of the naturally occurring product to sort of gauge the potential success of this product? Or is it just not enough available?
Well, that's a good question, because the natural compound is like our guru here, because we're seeing there is a fair bit of experiments that have been done with it, and we've learned a lot from it. And since the structures are so similar, we can confidently predict that whatever the flavopirin does, our molecule, NT1391, will do. And that gives us confidence. What's known about this naturally occurring molecule really applies. The research, even the papers published about flavo and liver cancer, will be true for our molecule. And so you have it's a leverage and an advantage to have this background in a natural compound, even if we did improve on it. I mean, it's
no doubt.
really, yeah.
So you don't have to answer this, but I'm looking at it as an investor, right? So if the natural occurring compound is safe, effective, is that you can't patent it, but is it like a nutraceutical, and so you can't be a drug, or it can be
That's
a drug?
right, exactly right.
Yeah,
that's right. It's marketed. You can market it as it is marketed as a dietary supplement.
yeah.
Yeah.
And is it $100 million a year business, or is it a $10 million a year business?
Oh no, it's much smaller. It's a small business. You can't. Is a limit. Availability is a real question there, because this is arduous. You have to go down to the Amazon. You have to have people you trust down there who harvest the bark.
Yeah.
Has to be shipped, and the quality control that's done is very impressive. It's treated very, very much like a drug. All the it's toxicology. It's all the tests for contaminants with heavy metals, bacteria, everything is done. Yeah. So it can be a safe product, but it's administered and it's developed and produced in small batches.
And are you at liberty to say, or do you have some ideas, like, has it been able to have people go from, hoping to use a year to hoping to be 5 years?
I think the answer is yes. And the way I put it is the use of that extract can extend life of cancer patients and maintain a higher quality of life. I think that's what you can say about it.
Yeah. And in the case of this drug, since you can manufacture a lot of it, and it I'm trying to use the right word, the high dosage is not toxic, then is this likely to be better in terms of, and I don't want to say curing cancer, but life's have a better lifestyle and live longer, that sort of, is that the best you can hope for?
Yes, I think we have here a breakthrough in cancer treatment, and it's that combination of safety and efficacy. It works against cancer, and we've allowed then, and it's safe, but we allow that you may, in certain cases, want to combine it with some drugs. And I should say those combinations there's some thought behind that and some data as well that shows that you get a dose reduction effect. For example, if you take our drug and combine it with a toxic chemo, or even a somewhat toxic chemo, you need less of the toxic drug to get an equivalent effect, because the presence of our drug supplements and synergizes really. So you can reduce the toxicity and the side effects, but you don't need to use as much of the standard mainstream drug and get the same outcome. But our drug seems to have this ability to restore, and this is worth saying, it restores the chemosensitivity of cancer cells to these drugs. And I didn't get a chance to say it, but many of the problems with cancer treatment are it's drug resistance. The cancer cell becomes resistant. It doesn't care if the drug's there anymore. It stops responding. So the drug stops having its effect. And in our preliminary data, it looks like presence of our drug together with the mainstream chemo drug overturns the drug resistance. And so this is been considered the holy grail for the next generation of cancer drugs. If you can restore chemosensitivity, is what it's called.
Yeah,
Yeah.
super, super interesting.
Yeah, thank you.
I really appreciate you sharing this with us. And it's just been, for me, it's a learning process from, ground zero. So thanks for bringing me up to the state of the art.
I tried. I hope I did.
Yeah, you certainly did.
Thanks.
So thank you for doing this. I'm gonna, if it's okay with you, we can do it again when things move along a little further. And of course, we're going to make sure everybody who RSVP'd will be in touch with you directly and see if you can have some meaningful conversations. So we're going to let everybody go now, but thank you for everybody for being here and allowing Family Office Insights and Dr. Hall to share this with you. Super, super interesting. Is way more fascinating than I expected it to be.
Oh, that's good.
Yeah.
Well, I look forward to hearing from anyone who has any questions or wants to talk about this more.
Yeah, thank you, Doctor, and thank you all for being here as usual and participating. And as I always say, thank you for sharing with us the only thing you can't make more of, and that's your time. Thank you, Doctor.
Thank you.
Okay,
Take care.
bye-bye.
Bye.